Hello, Namaskara. This is Pooja Joshi here again. I welcome you all to the NEET Zoology sessions. And we were discussing previously about digestion. That will be continuing even in today's class. Just before I finish the previous class, I mentioned a point that we shall discuss the questions based on the concepts that we have finished till then. But I think we will just move to all the questions at the end of the synopsis. Once all the explanation is over, we will move into the questionnaire. Alright? Yeah, so let's begin for today. In the previous class, we have discussed about nutrition. We have completed about components of food, which are nothing but the biomolecules that are necessary, that in turn break up in the body and ultimately produce energy. Then we also have gone through the digestive system, where we have covered about the alimentary canal. All right, we have understood that alimentary canal is a long tubular structure which measures to about 9 meters, and that is distinguished into various parts. Okay, the wall of the alimentary canal is almost uniform throughout the length of it, which is made up of 4 layers. All right, let just a small recap. The outer one is called as serosa. The outer one is called as the serosa or adventitia. Alright, I'll just quickly write the initials of it. The next one is called as muscularis, which includes the different arrangements of smooth muscles in the form of in circular form, longitudinal form, and in oblique form. Okay, so this way the arrangement of muscles makes about the muscularis layer. Inner to that is the submucosa layer. Okay, submucosa, inside which different glands at different parts of the alimentary canal are seen, and the innermost is the mucosa, where mostly the glands that produces mucus, which in turn helps in maintaining the lubrication of the alimentary canal, is seen. Alright, so this was then we saw that actual alimentary canal begins with the esophagus, okay, and ends with the anus. So what all comes in between that, you can just quickly look into, imagine your own digestive system. Starts with the esophagus, then we have the stomach, small intestine, large intestine, and finally the anus. Alright, so today we shall go ahead with the digestive glands, which equally, or rather more importantly, play a role, because these glands are going to decide the working of the digestive or the alimentary canal. Because without the action of the glands or the enzymes that the glands produce, the alimentary canal is just like a model in the sense, just like a showpiece. It has no role to play as such. Without, there is for its functionality, we need the glands as important as the digestive canal, and also most importantly in that, the enzymes that are produced by these glands. Okay, by saying so, we shall move ahead. This presentation was just to tell you what all we are still pending with. All right, yeah. So the digestive glands. Coming to the digestive glands, here I have enlisted the different types of glands that are present in the alimentary in the digestive system. First one is the salivary gland. All right, we all know that where these glands are present, we find them in the buccal cavity or inside the mouth or the mouth cavity. Alright, while studying the structure of it, we have seen that the buccal cavity consists of the teeth, the tongue, okay, the palate, and the cheeks on either sides, right? Yeah, so salivary glands, where exactly are they present? We shall go in detail with each one of this. The second set of glands, again as mentioned, esophagus does not have any glands per se. Okay, they only possess mucus producing glands, so that the bolus moves down the esophagus without much of a friction. Alright, so next glands are the gastric glands that are present in the stomach region. Alright, so we have these gastric glands embedded. Again, remember, whenever we talk about the glands in the alimentary canal, they are all present in the submucosa and the mucosa region of the histologically in the structure. Okay, so gastric glands are also present in the submucosa and mucosa regions of the stomach only. That's the term gastric given. Gastric term refers to stomach. Yeah, next gland is the liver. Liver actually is what do we say? It's a very multi multitasking organ rather. Understand one fact about liver. Liver is said to be the largest gland in the human body, and not just for digestion. Liver plays multiple roles, as I told you. It breaks down, of course it definitely helps in breaking down and metabolism of the biomolecules. Okay, it also helps in breaking down different types of medicines that we take, the drugs that are there, alcohol that is consumed by people. Okay, all of that. That's the reason, when they say a person is too alcoholic, he undergoes a liver disorder, because liver is the one which is overworking in metabolizing the contents of alcohol. Okay, so that's the reason. Not just there, even the dead RBCs, okay, some of the dead cells of the body, they come to the liver in order to get metabolized, and in such way liver plays multiple roles. Okay, now specifically with respect to digestion, what role liver plays is that it produces what we call as the bile. Okay, bile juice. What is this bile juice? We will come to that later. Okay, yeah, so liver is not exactly a part of alimentary canal like we find gastric glands. It is outside the alimentary canal, but it pours its secretions into the alimentary canal. When and where and how, we will come to that later. Yeah, pancreas is also similar to liver, as in it is not a part of the alimentary canal but situated outside. We find this pancreas located below the stomach, somewhere here, below the stomach and above the large intestine. Okay, so this pancreas is said to be a dual gland. Again, pancreas does not have the only role of in digestion. It also mainly plays role in endocrine system. Okay, pancreas is considered as a heterocrine gland. Heterocrine gland. What is the meaning of heterocrine gland? Heterocrine gland means it is endocrine in nature as well as exocrine in nature. Okay, so we have some idea about what endocrine and exocrine are. Okay, endocrine glands do not have any ducts. Whatever secretions are given out, those are directly given into the bloodstream, while exocrine glands have ducts that are connected to the bloodstream, to the target organ rather. Okay, so the secretions go through a channel called as the ducts in exocrine, but in endocrine they directly give out into the bloodstream. Okay, yeah, so when I say pancreas are both endocrine and exocrine in nature, also we can use the term as dual gland. Okay, the main role in the digestion is played by the exocrine glands, exocrine part of the pancreas. Next, moving on to the intestinal glands. Again, as the term says, intestinal glands refers to the glands that are present in the intestine. And when I say intestine, which is a part of the alimentary canal, they are embedded inside the submucosa and mucosa regions of the intestine. Okay, yeah, by saying so, we need to just quickly look at the different glands: salivary glands, gastric glands, liver, pancreas, and intestinal glands. Further, we shall be looking at what these glands produce, what are the functions of whatever they produce, okay, and ultimately how do they help in the process of digestion. Okay, yeah, so just quickly look at all these 5 glands. Okay, yeah, let's move ahead. Coming to the salivary glands. Okay, so salivary glands are 3 in number. Alright, so there are 3 pairs of salivary glands that are situated at different positions. One is situated just in front of the ear. We call this particular salivary gland as the parotid salivary gland. Okay, I'll just mention it as SG, salivary gland. They are a pair, which means that in front of one ear and the other ear, the other part of the pair will be. Next, coming to this one, which is called as the submaxillary or it's also called, another term is called as submandibular. In some books, it is referred to as submaxillary, in some other submandibular, but both are the same. This is sub refers to something which is below, right? Mandible is the jawbone. Okay, so mandible or maxilla, the jawbone, it is present beneath that. Okay, you can even touch there and just feel. You obviously won't feel it, but the places where they are present can be understood well. So submaxillary and submandibular salivary glands are the second pair, and the third pair are the sublingual glands. Okay. Lingua means tongue. That's why we say language, lingua. Alright. So tongue, language we speak with the help of tongue, right? Tongue helps in the formation of, twisting and turning, and then the sound production and the modification of the words is all because of tongue that helps us in do it. Okay, so hence sublingual salivary gland is also present in pair. So all these three pairs form the entire salivary gland system. All three almost produce similar composition. Okay, so what exactly do they contain? We will just have a look here. First and most important part is the mucus. See, throughout the alimentary canal, mucus plays a very, very important role. Alright, because it acts as a lubricant, one thing. Second, it helps in maintaining the pH. pH in the sense, sometimes the enzymes whatever are produced have a pH which is not in the range of normalcy. Okay, so such mucus plays a major role in maintaining the pH, okay, and mainly, as I told you, in lubrication of whatever is consumed, and so that it easily passes through the alimentary canal. Yeah, as I told you, it serves as a lubricant. Next, amylase, salivary amylase. Remember, amylase, amylo, this term is used in reference to the carbohydrates only. Okay. So, amylase, amylo, whatever you find, the term amyl, wherever this suffix of amyl is used, it refers to carbohydrates. Alright, so this amylase initiates the digestion of starch. Whatever food we consume, remember the carbohydrates that we consume in the form of, let's say, potato or rice, they are all complex carbohydrates, also categorized and named as starch. So starch needs, the digestion of carbohydrates partially begins in the mouth itself. Okay, you all must have heard everybody at some point of time saying that you must chew your food 32 times. What is the logic behind this? It's very important when it, even from the point of view of medical sciences or Ayurveda, it says that we are providing it time, okay, for the food to properly get digested, and so that the action of amylase properly takes place. I will give you two situations. One thing, you are in a competition where you are made to eat food properly. Okay, if you do any kind of hurry burry there, then you lose the competition. Okay, that is one. Second is where you're supposed to eat fast. All right, so what happens when you're going to eat fast? What do you tend to do? Whatever is there in front of you, you're just going to put it in your mouth, chew it for about 1 or 2 times and swallow it. Okay, because you want to win the competition. That's how most of us eat food these days. We barely have time for anything, so we do not focus on what we are eating, and we simply put things in our mouth, chew it once or twice and just gulp it down our throat. This is not the way. While the other competition that I told you, where you're supposed to eat food chewing it about 25 to 30 times, what happens to the body? Actually, that is the most ideal way. These days, we people do not have time, so as I told you, we simply just stuff food in our mouth, but that is absolutely a wrong way, and that will lead to further complications. Let us see what happens when you chew food properly. Okay, mucus properly acts, amylase enzyme acts. Let us say you are just consuming rice, a morsel of rice. Okay, so to some extent the starch already gets digested here, because you're going to chew it and keep it in your buccal cavity for quite a long time. Okay, so what happens is each and every molecule of starch gets broken down or gets acted upon by amylase, and it is converted into its next form, that is a maltose. Okay, so the chemical reaction and all that we shall see in the later slides. But remember that starch polysaccharide gets converted into disaccharide or any oligosaccharide, but mostly into maltose. Okay, and now next what you are going to do is you are going to send this maltose into the next process of digestion. Okay, now wherever next part this carbohydrate is going to get digested, this disaccharide will get converted into the monosaccharide, that is the glucose. Okay, and the intestine properly absorbs glucose, and each and every biomolecule has similar pattern of working. But what we do, in case you just eat rice and because it is soft, if you just swallow it, there is no time for starch to get digested in the buccal cavity. It will go as starch in the stomach, let us say. Okay, in stomach or in whatever region further it gets digested, instead of maltose it has starch only, which is still not got digested. So the enzyme, let us say pancreatic amylase, will act on starch, okay, instead of acting on maltose, and now this starch will form into maltose, where instead of mouth this is forming into maltose in the intestine. Then furthermore, this maltose barely gets any time to form into glucose. Okay, maltose form is not properly absorbed by the body. Okay, most of it, I'm not saying entirely, some part breaks, but not in efficient one. Okay, so whatever nutrition you have consumed goes out in the form of maltose itself. This is just an example to make you understand the importance of process of digestion starts in the mouth, so we need to focus on that as well. Okay, simply eating for the namesake to fill the stomach is going to further lead to various other health issues related to digestion. Okay, that's the reason we people at the age of initial 30s and 40s, we undergo hyperacidity issues, we are obese. All right, there are so many other health issues, that is just because of the way we eat food. All right, so this is a very important thing to ponder on. Okay, so yeah, let's just go ahead. Lingual lipase. Okay, lipase, lingua. Lipase, that is some very meager little amount of lipase is produced in the buccal cavity, and that is a part of salivary gland that acts on the fat to some extent. Okay, not as much as starch. Next, we have electrolyte solution that includes sodium ions, calcium ions, potassium ions, okay, and bicarbonates. All these inorganic ions or electrolytes help in mixing of the food, okay, and moistening of the food, and ultimately they help in formation of the bolus. All right, next component, proteins and the enzymes. Okay, statins, proline rich proteins, histamines, okay, then lysozymes, salivary peroxidase. All these, lysozymes especially, we hear most of the time about this, and salivary peroxidase. So both of these, what they do is they have antibacterial or antimicrobial action. Any bacterium, like as I told you, suppose you are going to eat something on the roadside, pani puri wala, okay, we know how hygienic they are, right? So maybe there are some, maybe no, definitely there will be so many microorganisms associated with what we eat on the roadside. So but still our system is going to take care of it to some extent. Okay, so whatever microorganisms are there that can be easily available and easily killed in the buccal cavity, and again if you give time by chewing properly in the buccal cavity, then the microbial action also will be done, and almost dead bacteria only are sent into the stomach, which obviously will not cause any harm to you because they will be thrown out as waste products. Then again, all these enzymes help in lubrication and buffer capacity and remineralization. Okay, so remineralization in the sense, whatever food to some extent, minerals what we consume through food, addition of minerals is done through these inorganic minerals, as I told you, sodium, calcium. Electrolytes are nothing but inorganic elements. Okay, buffer capacity, again buffer means something that maintains the pH. Okay, so remember, to maintain the pH also, all of these components of salivary glands play a major role. I hope salivary gland concept was clear. Alright, yeah. Coming now to the gastric glands. Okay, now food, whatever is, as I told you, if you are going to eat food like the first case example what I told you, eating it, chewing it properly, allowing the enzymes, mucus, and all of the buffer action to take place, then okay, you swallow the food, or kind of deglutination takes place. Then the food further moves down the esophagus and reaches the stomach. So let's imagine this is the food that has reached your stomach. See here, there are two sphincters. Just recall what the names of the sphincters are. This is the cardiac sphincter, okay, and this is the pyloric sphincter. Okay, so food when it comes here simply doesn't drop down. There is constriction which needs to be opened, and that happens by the wave like motion that comes. Okay, so the wave that expands pushes the food into the stomach. As I mentioned in the previous classes also, I told you that by the time you're even thinking of food, the secretions throughout the alimentary canal already begins. So the moment you eat food, the stomach already has this much amount of fluids that are poured into the lumen. Okay, this is to keep this is the get set go kind of preparation that the stomach does, and that when the food enters inside, direct breaking down of it begins, not all of a sudden, but over a period of 4 to 5 hours, as I mentioned. Okay, for 4 to 5 hours, food will be, when it enters, it is in this form. You can see that it is gradually getting broken down, and by the end of 4 to 5 hours, we do not find complete food at all, or the bolus at all. They are all in the form of broken elements here and there. Okay, yeah. So what do these gastric glands do? Okay, water further liquefies the swallowed food. All right, remember, bolus is a semi-solid structure. Next after that is the chyme. chyme, chyme. Chyme is a semi-liquid, or rather more of it is liquid content. Okay, so bolus is in the buccal cavity, chyme is in the stomach. What you see, the yellow color structure here, is nothing but the chyme. Alright, so water is also produced by all of these glands, and that water is going to help in further liquefaction of the food. Alright, hydrochloric acid. This acid, remember, hydrochloric acid produced in your own stomach, it is so strong that it has the capacity to completely melt a blade. Okay, the 7 o'clock blade that is used for shaving, it has the capacity to melt it over a period of an hour. So just imagine how strong the acid is that is produced. And we all know how dangerous acids are, what they cause to the skin. Just imagine stomach, entire alimentary canal being made up of soft or smooth muscles. It would have completely got destroyed just by some drops of hydrochloric acid, but still it will be maintained till the end of our life, right? The digestive system. This is because of the mucus. We need to really applaud the role of what the mucus plays. Such strong acid, though it is being produced by your own body, it does not cause harm to your own self, unless and until your food habits are really bad. Okay, if you eat highly acidic food, foods that trigger more acid production, then if it is beyond its limit, then the acid ulcers, stomach ulcers and all that begin. Alright, so what that causes, we will come to that in the disorders. Alright, yeah, so first, the role of acid, it is dangerous, but to our system, what role it plays? It acidifies the food and stops the action of salivary amylase. Okay, till the bolus contains the salivary amylase, that action is stopped in the stomach by the hydrochloric acid. Next is, it kills ingested microbes. In case some microbes have escaped the lysozyme that is produced in this buccal cavity, those are all killed in the stomach because of the hyperactivity of hydrochloric acid. Okay, then provides the acid environment needed for action of pepsins. Remember, pepsinogen is an inactive enzyme which is converted into active enzyme called the pepsins in the presence of HCl. Okay, hydrochloric acid. Without the presence of HCl, the conversion of inactive enzymes does not take place, and if that does not happen, the enzymes that are supposed to act on the protein part of food will not take place. Alright, yeah. Next is, gastric glands also contain pepsinogens, as I told you. These are inactive enzymes that digest proteins. Intrinsic factor. Intrinsic factor is an interesting actually content. Actually, its role is not in it doesn't work directly on the major macromolecules. What I told you. What are the macromolecules? Just recollect. Carbohydrates, proteins, and lipids. Exactly. Then what does it act on? It acts on the vitamins, specifically vitamin B12. Vitamin B12 is actually all vitamins are equally important, but vitamin B12 needs a type of, it has to be converted or joined with something and then absorbed by the body. Just as vitamin B12, it is not absorbed. So these intrinsic factors that are produced as a part of gastric secretion, they combine with vitamin B12 and then finally get absorbed. Okay, so remember the role of intrinsic factor. Finally, the mucus, as I told you, very important, omnipotent content throughout the alimentary canal, plays a very important role as well. I hope I'm clear what the secretions of gastric glands are. Okay, just quickly read what the slide says for a minute. Fine, let's go ahead now. Yeah, an image to show you how the arrangement of these glands are seen in the gastric in the stomach. Just a piece of stomach image vertically. Suppose this is the stomach. Okay, very bad shape, I know. Yeah, just the wall of it, maybe this section. This is the image of that. Okay, so yeah, these are called as gastric pits. As I told you, the secretions are given from submucosa and mucosa regions. Those are present here. Muscularis, submucosa. Okay, this region, the pits that are there, they are all lined by specialized cells, epithelial cells that produce the required secretions. All right. So let's just look at this. Internally, in the base of these pits, we have the gastric glands. You can see here, the black region what I'm drawing here. Okay, these are the gastric glands. And what exactly are produced here? The epithelial cells, surface mucous cells. They are also called as the goblet cells. Goblet cells. Okay. Then we have mucous neck cells. Both of them secrete mucus. These are different types of cells but grouped under goblet cells. Commonly goblet cells produce mucus. Then we have the parietal cells, which are located here, the ones with wider bigger nucleus. They produce HCl and the intrinsic factor. Chief cells, which produce parietal cells, I forgot to mention, are also called as oxantic cells. Oxantic cells. So in case they ask you, they give you a question saying that which of the following cells of the stomach produce HCl, and the options given are goblet cells, chief cells, G cells, and oxantic cells. So you should not get confused, because oxantic and parietal are different names of the same cells. Next, coming to the chief cells, they secrete inactive pepsin or pepsinogen and the gastric lipase. G cells secrete the hormone gastrin. Now, what role this gastrin plays, we will just remember. Okay, do not forget what these G cells are. They the role of theirs will be studied in endocrine chapter. All right. So gastrin, G cells. So what are the four types of cells? Goblet cells, oxantic or parietal cells, chief cells, and the G cells. Okay. Yeah. Next. So I just pardon me for the blur image of it, but I hope things are understandable. Yeah, so gastric juice produces water. 99.5% of it is water itself. Solids, which are 0.5%. Then solids are of two types. You have organic substances and inorganic substances. Under organic, we have enzymes and other organic substances. So just have a look at what are the enzymes, types of enzymes. Pepsin, renin. Renin is found only in animals. Okay. Then we have gastric lipase, gelatinase, and urease. In other, we have mucus and the intrinsic factor. These are not enzymes, but they are again the organic components produced by that are a part of gastric juice. Next we have the inorganic substances, which are HCl, sodium ions, calcium ions, potassium, bicarbonates, chlorides, phosphates and sulfates. Okay, all of these again they form the chyme. Remember, chyme is completely liquid. And why it is liquid? Because so much percent of it, when it is water, definitely the content is going to be liquid itself, right? And suspended in them are all these molecules. Yeah, coming to the next one, that is the reactions. See, I am just explaining you what they produce, what are the actions and the reactions, biological reactions. We shall go to that later. Yes, so liver, what role does liver play? I already mentioned in the initial slide that liver produces what we call as the bile juice. Okay, so bile is made in liver. And bile, though it is made in liver, I told you that liver already has so many things to do. So it does not have place to store bile. What it does, it sends the bile to something called as a gallbladder. Gallbladder is, it is also pronounced as gallbladder or gallbladder, anything of it, it's fine, unless you write the spelling correctly. Yeah, so the gallbladder is closely associated with the structure of the liver. It is outside the liver, okay, and it is a small muscular pouch that stores whatever bile is produced. As and how it is produced, it comes and fills into the gallbladder. This gallbladder does the work of how the urinary bladder functions in our body. As and how urine is produced, it gets stored in the urinary bladder. Okay, similarly, this is the function of gallbladder. Next, what does it help us with? It helps us with digestion, specifically of fats. Okay, absorption of fats, that is, it emulsifies and solubilizes fats. Understand that fats are not soluble in water. This is a known fact. If you mix oil or ghee or any wax in water, does it mix with it? No. So it is insoluble. But some way or the other, it has to be absorbed, right? So bile, contents of the bile help in solubilizing, or rather emulsifying the fats and absorbing it. Proteins and carbohydrates have no problem. They mix with the water and they are absorbed. Next, what else does the bile help with is excretion. Okay, see, bile here I'll take a little time extra in telling you about bile and liver because it is really important. You remember I mentioned a point saying that breakdown of many cells, okay, especially the cells that are going to wear out and tear out, that happens in the liver. Liver is kind of like, a mortuary where finally the things are completely broken down of the dead ones and they are they sent out of the liver. Yeah. So RBCs, we all know when you study the cell chapter, it is taught to you that all cells do not have a same lifespan. Okay, the cells that were present in your body when you were born, most of them are not present right now because lifespan of every cell is different. RBCs likewise is 120 days or 40 or 4 months, right? So what happens? What about the old ones? As and how the cells get old, they come and they enter the liver. The enzymes of the liver, what they do is they break down the RBCs bit by bit. Okay, the contents of the RBCs are all separated and they are sent through to get excreted out of the body, because the contents of the dead RBCs actually have no role to play in the body. So body knows very well when whatever is not going to contribute for the proper functioning of the body, it is going to throw it out. Alright, likewise, the pigments, bile pigments, what we say, bilirubin and biliverdin are products of the metabolism of the RBC. Alright, a big breakdown of RBC. Those colored components, because red color hemoglobin is there. Once the cell wears out, it is completely going to degenerate. Then the color becomes kind of yellowish or greenish, and that is what is the bilirubin and biliverdin. Okay, bile pigments, what is mentioned here, they are a part of the bile content. Okay, yeah. Then for the process of absorption of fats, they are the bile salts. Okay, sodium taurocholate is one such which is a salt that helps in emulsifying the fat. What is emulsification? Large fat globule is converted into pieces. Okay, by disturbing it. Okay, by disturbing it, it forms into various small pieces. so that the lipase enzyme can act. Lipase enzyme, understand, can never act directly on the large globule. Even if it acts, it will completely never digest it. Okay, maybe some surroundings, it actually doesn't happen in that way. So first the bile salts have to act on it such that the pieces of it, it breaks it completely, and on these individual pieces or emulsified products of the the large lipid, the lipase enzyme acts and completely forms into its monomers. Okay, yeah. So bile composition of the bile, as mentioned, it has got bile salts, phospholipids, cholesterol, and bile pigments. Okay, though bile pigments are in smaller percentage, the coloration of your fecal matter is because of bile pigments, because through the fecal matter the excretion of bile pigment takes place, and to some extent bile pigments are also part of your urine. That's the reason the tingy yellow color of your urine is because of presence of bile. Okay, by saying so, we shall go ahead to the next slide. Yeah, another image to just give you an idea that how the gallbladder is and how the duct is. Okay, from different parts of the liver, bile is connected and then poured into the gallbladder. Alright, further, common bile duct is going to carry the bile from the gallbladder and release it into the small intestine. Okay, yeah. Next one. Moving on ahead to the pancreas. As mentioned, pancreas is a heterocrine gland or the dual gland. Okay, just have a close look at this. Pancreatic islets of Langerhans. Okay, so these islets are smaller than islands. Okay, and they appear as small scattered groups here and there. So when you look into the histology of pancreas, you will find accumulated cells are scattered here and there. Okay, so one of them, you can see these blue ones. So blue ones represent the islets of Langerhans, and they produce the hormones, which is the endocrine part of the pancreas. Okay, now what are the exocrine? I told you exocrine have glands, right? So they are called as the acinar cells. Acinar cells are also present equally scattered everywhere, more in number when compared to the islets. Okay, and each acina is connected. You can see the white color hole here, right? That is nothing but the opening of the duct. Okay, and the green color that you see, just a few of them have been represented. So they pour whatever secretions these cells give. Blue color are the nucleus. Okay, nuclei of each cell. Whatever secretions they give, they are poured and they are sent along the direction of the duct. Okay, so here you can see pancreatic duct. Finally, small clumps of acinar cells here and there, they are connected here. Okay, like this is the main highway, pancreatic duct, you can consider that as a highway. Various state highways get connected to it, and finally whatever secretion is produced is going to open into the duodenum. Here you can see the bile duct opening into the duodenum. Alright, because liver and pancreas do not have an axis. They are not a part of alimentary canal. That's why you cannot they cannot come directly in touch with the food or the chyme, right? So they have to pour the secretions to a part where the food is present. So that happens in the duodenum region. Okay, so just have a look at the different parts of it. Yeah, let's go ahead. What is the composition of the pancreatic juice? Again, remember all the secretions, most of them have water. So 99.5% again, pancreatic juice all contains water, 0.5% of solids. Solids are into inorganic and organic. Organic contain enzymes, which are, please pay attention here, the enzymes of the pancreas. You have trypsin, chymotrypsin, carboxypeptidase, okay, yeah, collagenase, nuclease, elastinase. These are all proteolytic enzymes. Proteolytic, the term itself says they digest the proteins. So until now we had seen only one enzyme produced in the gastric gland called as the pepsin, okay, which digests the proteins, so proteolytic enzyme. Next to that, it is a pancreas which produces 6 different enzymes to digest proteins. So just see what varieties of proteins are present which need digestion separately, right? Yeah. Next one is amylolytic enzyme, pancreatic amylase. This is the only enzyme, again, everywhere it's only the amylase that digests the carbohydrates. So because it is produced by the pancreas, it is a pancreatic amylase. Coming to the lipid digestion, what all they produce? Pancreatic lipase, cholesterol ester hydrolase, phospholipase A, phospholipase B, colipase, bile salt activated lipase. Okay, so these are the enzymes produced by the pancreatic juice. Just see the small gland, so much it works, right? Other organic components are again albumin and globulin. These are two types of proteins itself which the body produces, all right, which the pancreas produces rather. Inorganic, again, all these electrolyte components, inorganic substances like the sodium, calcium, potassium, magnesium, bicarbonate, chloride, phosphate, sulfate, all these are parts of the inorganic contents. All right, moving ahead. Pancreatic juices. All right, so amylase converts, just to see what all it does quickly. Amylase converts starch into maltose. Again, I told you, in case starch properly gets digested in the buccal cavity, then you directly have maltose to be acted upon, right? But nevertheless, some amount of starch is also converted by the pancreatic amylase. Enterokinase converts trypsinogen into trypsin. Trypsin is the protein. Inactive form of that is trypsinogen, which enterokinase converts. Enterokinase is actually produced by the intestine in the duodenum. Trypsin converts proteins into dipeptides. We have polypeptides which are broken down by the trypsin, converts it into dipeptides. Trypsin also converts chymotrypsinogen into chymotrypsin. Okay? Yeah. Chymotrypsin converts peptones into dipeptides again. So we have polypeptides, peptones. These are different forms of structures of proteins. All right. Finally, they need to get converted into dipeptides so that the dipeptides are broken into amino acids. Trypsin also converts pro-carboxypeptidase into carboxypeptidase. Now, peptidase, anything that ends with ASE is an enzyme. So this is still an enzyme, and carboxypeptidase now converts proteoses into dipeptides. Trypsin converts proelastase into elastase. This is again an enzyme. So just see, trypsin is a precursor. Rather than directly acting on the proteins, it is going to activate. It is like, rather you just wake up the first person of the house and then he goes on waking up all the others. It's somewhat like that. So trypsin, you are going to wake this up. Okay. So furthermore, like the enterokinase is going to do that work, and trypsin is then going to wake all the other enzymes to carry out the further processes. So elastase now converts elastin into dipeptides. Okay, elastin also is a type of protein. Pancreatic amylase converts polysaccharides into disaccharides, like the maltose if it is starch. Nuclease is nuclease is in the pancreatic juice, acts on the nucleic acids and forms nucleosides and nucleotides. In detail, I'm sure about this. You might have studied about the structure of the nucleus, the nucleotides in your 12th in molecular basis of inheritance, or even in biomolecules, I suppose you have studied about this. Yeah. So now you may wonder, from where are nucleic acids going to enter your body? You are going to consume food which is in the form of cells. Like, if you just let us just consider you are going to consume a leaf of spinach. Spinach is a leaf. Leaf is made up of cells, right? So the cells contain the nucleus. Nucleus contains the they contain the nucleotides and the nucleic acids. Okay, so those nucleic acids also need to be broken down, and nucleases does this function. Okay, now these nucleotides and nucleosides which are there, they will act as raw materials in the formation of new strand of DNA. We'll come to that later. Okay, so just quickly have a look. If possible, just jot this down in short form because it's a very important slide. I'll give you a minute. Yeah. So let's move ahead. This is another image that gives you the exact track of things, how they move. From liver, bile is produced and stored in the gallbladder. As and when required, the gallbladder releases its secretions and open into the duodenum. Okay, stomach, the acid chyme comes through the duodenum. Pancreatic juice is also produced and then poured into the duodenum. So duodenum is like a common connection point where the chyme, bile and the pancreatic juice meet. Not just that, furthermore throughout the duodenum, even the intestinal juices are produced. Okay, on so that they act on the chyme. Coming to the intestinal glands now. Intestinal glands, you can see these faint structures. They are the villi. Okay, one such villus is extended here. Okay, is represented here. They are lined by different colored cells. Let's just see what are the roles of the cells that they play. Cells that are present. See, one thing, in case of intestine, we have deep structures rooted inside the villi. We call those as crypts of Liberkun. Okay, crypts of Liberkun or Liberkun, whatever way it is pronounced. These crypts mean something which is dug and which is deep into the ground. This is how it appears. Okay, so inside these crypts, what part you see here, that is the intestinal gland, and what all it produces, we shall look at that now. First is the intestinal stem cells. Look at these, the red color ones that you see, these are the intestinal stem cells. I'm sure you are all aware what role of a stem cell is. It helps in regeneration of new cells, right? As in how the intestinal cells wear and tear, the stem cells help in producing new cells that modify and become whatever type of cell is necessary. Then we have Paneth cells. Paneth cells also produce mucus that help in lubricating. Okay, we also have goblet cells, which again produce mucus, the yellow colored cells that you see. Then we have transit amplifying cells. Then we have enteroendocrine cells, the green ones. Okay, then enterocytes are nothing but the intestinal cells, epithelial cells. Okay, yeah. The content, what we say, intestinal juice, it has a proper name given called as the succus entericus, which is nothing but intestinal juice. So this also has 99.5% of water, solids, organic substances, inorganic substances will straight away move to the enzymes that are there, that are produced by the intestinal juice. We have different peptidases, aminopeptidase, dipeptidase, tripeptidase. Now, these peptidases act on the amino acids, finally giving end product should be amino acid now. Okay, dipeptidase, they act on the dipeptides to produce amino acid. These act on the tripeptides to produce amino acids. We have enterokinase, okay, and lipolytic enzyme, which is lipase, acts on the broken lipids, finally giving out fatty acids and glycerol. Amylo ones, we have sucrase, which acts on sucrose and produces the monosaccharides. Okay, we have the sucrose is glucose and fructose. Maltose is acted upon by maltase to produce glucose. Lactase is acted upon by, is going to act on lactose. Dextrinase is going to act on dextrin. Okay, 3-halase is going to act on 3-halose. Okay, so next we have mucus again. Mucus and intrinsic factors are common. Defensins are again other substances that help in protecting the alimentary, the intestine from any potent or potential microorganism or a pathogen that has entered and that has successfully escaped the lysozyme and the HCl. Alright, like such things are especially which are in cyst form. Cyst is nothing but a hard covering which the HCl and also the lysozyme cannot break them. The young one or the organism is present inside the cyst. Okay, so even at intestine level we have defensins which help in that function. Common electrolytes are again part of the structure. So digestion in the buccal cavity. sorry for the yeah. So digestion in the buccal cavity and stomach. So this is just going to give you an overview of what all we have studied till now. So please pay attention and look at it. Okay. What all happens? Mechanism of digestion. Yeah. So in the oral cavity, when we talk about the oral cavity, your mouth region, food is taken through the mouth. Teeth break down the food mechanically. The saliva, which is a combination of amylase and lysozyme, they convert 30% of starch into the disaccharide. Antibacterial agent lysozyme is an antibacterial agent that helps in preventing potential infections. Okay. Here what else we do is we by chewing, food is broken down into small particles and forms a bolus. Saliva moistens and lubricates food, forming a bolus. Bolus is then swallowed and moves through the esophagus by peristalsis. Peristalsis is a rhythmic contraction and relaxation of esophageal wall. Starch plus salivary amylase produces the disaccharide maltose. Moving on to what happens in the stomach. Gastric glands in mucosa lining. So we have mucous neck cells that produces mucus, peptic cells that produces pepsinogen, parietal or oxantic cells that produces HCl. So food mixes with the gastric juices by churning movement. Churning is nothing but continuous, somehow like, when you're mixing something with the help of a spoon, okay, something in a large vessel, when you're trying to just mix everything and the contents are really thick and you're putting energy to mix that is called as churning, or stirring rather. Continuous up and down movement is also called as churning. So chyme is what is formed. It is a semi-digested, acidic and pulpy food. So the semi-solid bolus is formed into semi-liquid chyme. It's also called as chyme, but mostly it is pronounced as chyme. So gastric juice and enzyme. HCl provides acidic pH. Pepsinogen, a proenzyme, is converted into pepsin by HCl. Pepsin converts protein into peptones and proteoses. Prorenin, which is a proenzyme, is converted into renin by HCl. This is in case of animals only, not in humans. Casein is a milk protein converted into peptides by renin. Alright? Yeah. So, I want you people to again look at it for some seconds. Alright. Digestion in the small intestine and the large intestine. So in the small intestine, you can see how clumsily arranged this structure is. You know why it is so? This is so that the food is held inside the intestine for as long as possible, so that complete digestion takes place. Once the food is inside, the pushing movement, peristalsis, is actually seen throughout the alimentary canal. Only because of peristalsis, food is pushed ahead. There is nobody else sitting there and thumping food and pushing it ahead. It is only the peristalsis that helps in the movement of food. So once the food reaches, it is involuntary. It is also not under your will and wish. So it is going to keep moving further. Imagine if the esophagus was just a small, tiny, if it was a small pipe or it was small in length. Complete digestion of food would definitely not have occurred. Okay, there is another interesting point I would like to mention here. The alimentary canal, rather the small intestine length of herbivores, is very, very long when compared to the small intestine length of the carnivores. This is because herbivores consume raw food, which needs a lot of time to get digested and broken down. As I mentioned, appendix, which is there in these herbivores, produces cellulase, which takes time to digest the cellulose. That's a part of the plant cell wall. When it comes to carnivores, we all know what carnivores eat. They eat the flesh, ready-made protein, muscles and flesh of another animal. So that protein does not need a lot of time to get digested because it is already in partially digested form. Okay, so to convert that and get absorbed, it does not take much of a time. So when you compare the length of a herbivore small intestine and a carnivore small intestine, herbivores is longer than the carnivores. Okay, this is just for your information. Yeah, small intestine consists of duodenum, jejunum and the ileum. Chyme is further mixed and churned by peristalsis. Pancreas and liver secrete enzymes that help break down and absorb food into the blood. Absorbed nutrients are assimilated upon reaching the target. Okay, like I told you, imagine your fingertips. Okay, you when or any baby, you know how it grows. So that whatever is consumed goes and reaches each and every target organ, and such that it results in growth and development. The digestive juices and enzymes. Pancreatic juices break down carbohydrates and proteins. Intestinal juices break down carbohydrates and proteins. Bile converts fat globules into fat droplets by emulsification. Pancreatic lipase converts triglycerides into fatty acids and glycerol. This is very important. Have a look. Yeah. Large intestine, significantly less digestive activity takes place in this. Mostly it results in absorption of water. So bacteria act on leftover food particles. Okay, absorption of minerals, water and certain drugs takes place in the large intestine. Large intestine secretes mucus that helps in lubrication and holding up of waste particles. Undigested and unabsorbed fecal matter are passed into the rectum. Fecus or feces is digested through the anus. Okay, so you know this structure what in yesterday's diagram we studied. Sorry, the cecum to which the appendix is attached. Here we find abundantly the rich source of friendly bacteria. These bacteria are going to consume the food that whatever enters into the large intestine for their activity, and as a result of reactions that happen in the bacteria, a gas is produced. Okay, so this gas is somewhat similar to H2S2. Okay, so that gas accumulation happens in the food. You know, the fecal matter smells because of the work done by the bacteria present in the large intestine. Okay, yeah. So saying so, again I'll give you a few seconds to just look at this entire slide. Yeah. Okay, another image that will give you a proper insight about how things work. Carbohydrate digestion. Please follow me if you have not paid attention previously. Oral cavity, pharynx and esophagus. So polysaccharides, disaccharides, salivary amylase converts them. The polysaccharides are converted into smaller polysaccharides by salivary amylase. Okay, so now this disaccharides are not digested there. Disaccharides, in case you've consumed any form of disaccharide, like if you've consumed maltase for example, that does not get digested here because it directly moves to the small intestine. Its action takes place in the small intestine. Maltose that is formed by the digestion of any kind of polysaccharide, that also gets worked upon by in the small intestine. Stomach, no digestion of carbohydrate takes place. Okay, small intestine enzymes. Okay, we have pancreatic amylase that acts on the disaccharides and then it is pushed further. So it's in the small intestine at the end where the monosaccharides are formed. Coming to protein. Protein digestion does not take place in the buccal cavity. Okay, so in stomach it starts. So proteins that are there, complex polypeptides or proteins, are converted into small polypeptides by pepsin. In the small intestine, initial part, the duodenum, pancreatic trypsin and chymotrypsin convert smaller polypeptides into even smaller peptides. Okay, pancreatic carboxypeptidase, they form into amino acids. Smaller peptides are acted upon by dipeptidases, carboxypeptidases and aminopeptidase, ultimately giving you the end product, the monomer, that is the amino acid. Digestion of fats does not, it's very negligible, so we'll just say that it does not take place in the buccal cavity, nor does it take place in the stomach. Okay, I'm sorry, nucleic acid. So the DNA and RNA that enter our body through different kinds of food materials that we eat, they are converted into nucleotides by pancreatic nucleases. So nucleotidases converts nucleosides into nitrogenous bases, sugars and phosphates when they are acted upon by nucleosidases and phosphatases. Coming to fat digestion, similarly, it does not take place in the buccal cavity nor in the stomach. Though there is meager amount of lipase present, because the emulsification has not yet occurred, there is partially or barely any kind of digestion of complex lipids that takes place. Finally, when we come to the small intestine, the fat triglycerides are converted into glycerol, fatty acids and monoglycerides by pancreatic lipase once they are in triglyceride form, which is done by the bile salts. Okay, yeah. So in today's class we shall just end it here. In the next session we shall quickly go through the reactions of these biomolecules. Okay, yeah, and then we shall move on to the questionnaire. We'll quickly go through the disorders of the digestive system as well, and finally go to the questionnaire and try to understand how much we have understood from whatever has been explained, and try to crack the questions of zoology digestion chapter. Okay, yeah, by saying so, thank you for today.